Last week, I talked about parabiosis, which might be disturbing for some people. This time, I'm gonna talk about something related, but not as gruesome as parabiosis. So, let me broad up the question. What exactly is chimerism? By definition, chimerism, or also known as genetic chimera, is a condition where an individual has cells with more than one unique genotype. You might also know about hybrid, where two organisms of different clade crossbreed. Hybrids also have two different genetic origin. The difference is, for hybrids, all of their cells basically have the same genotype. Meanwhile, chimerism means they have at least two sets of cells, each set with distinct genotypes. So no, breeding two different species of animals does not result in chimerism. You have to do some specific things for that. However, chimerisms do happen naturally. While it is especially prevalent in protists and plants, it also occurs in the animal kingdom. So let's talk about that. So, how could it happen naturally? There are at least two known ways. The first one is, quite simply, fusion. Some animals could fuse naturally. For example, the recently published article, where comb jellies are observed to fuse together. In this case, their digestive and nervous system are also integrated, so it's kinda similar to the parabiosis that I talked about last week, but it happened naturally. In such case, each site will retain their cells with distinct genotype, so it resulted in chimerism. Natural fusion doesn't just happen in basal groups of animals. One famous example of this is the case of deep-sea anglerfish. Males will embed themselves to females and be integrated. In some cases, multiple males could embed themselves into one female. Female of triple-wart sea devil, Cryptopsaurus coesi, had been observed to fuse with 8 males, so that's quite something. Chimerism is also prevalent in colonial animals, like the colonial hydrozoan. Some colonial hydrozoans do produce its own bud asexually, but some of them aggregate from different individual polyps. In this example, juvenile polyps settle into the colonial adult, and then after some time they are completely fused. In this case, they will exhibit chimerism. Chimerism was also detected in sponge. We don't have a definite answer to how and why. A possible explanation is a larva that is trying to embed themselves to substrate turns up embedding themselves to a pre-existing sponge instead. Another possible explanation is a case where a larva failed to be released from the adult, and then it developed within the mother. Another case of naturally happening chimerism happened in the reproductive or developmental stage. A commonly known example is the chimerism of marmosets. So, marmosets usually produce twins. They not only share womb, they also share a circulatory system from their mother. That's why, sometimes, stem cells from each other flow into the other. That stem cells then become blood and immune cells. And that's why, marmosets usually have some cells originating from their twin. A different case occurs for the half-sided Budgerigar. In this case, they were originally twins, but during development, the two embryos fused, so it hatched as one individual with two sets of cells, one from each embryo. Another example of this is the half-sided axolotls. A relatively recent and interesting finding is the case of obligate chimerism in yellow crazy ant. So, hymenopterans, that is ants and bees, usually have haplodiploidy sex determination system, perhaps more commonly known as XXXO. Haploids, aka XO, are males, that is, the drone. Meanwhile, diploids, aka XX, are females. Normally, they become workers, but if they are fed a lot of food during development, they will become the queen. However, this is not the case for the yellow crazy ant. In yellow crazy ant, queens are genetically distinct from workers. Queens are notated as RR, and workers are notated as RW. Both are diploids. Now, that sounds weird if you know how asexual reproduction works. Let me walk you through it. Workers are not sexually reproductive. Only queens do so. And queens are RR. Now, the gamete would be R and R, aka the same. One of these will be fertilized by male's gamete. Now, remember, queens are RR and workers are RW. So males will somehow have R genotype and W genotype at the same time. So they should be RW. But that's weird. If it is indeed RW, that would be the same with the workers. How could that be? But no, that is not the case. After several testing, it is shown that they don't have RW genotype. All of the males are haploids, but they have some R cells and some W cells. That is chimerism. Because of this condition, some of them produce sperms with R genotype, while some produce sperms with W genotype. Now, let's talk about the artificial chimerism. But before that, artificial chimerism is a chimerism artificially made by humans. That's quite obvious, isn't it? This was mostly done in the medical field, for organ transplantation research, regeneration research, genome editing research, stuff like that. The way to artificially induce chimerism varies. For animals that could naturally regenerate themselves, they sometimes wound two individuals, put them close to each other, and let them regenerate. Sometimes, they could accidentally fuse together during their regeneration. Well, quote-unquote accidentally, I guess. One of the methods for artificial chimerism, an infamous one for animal, is grafting. Those who like gardening or just plants in general might be familiar with grafting. Grafting is a technique where tissues of two different plants are joined together and continue growing as one individual. And yes, you could also do that to animals, and it had been done numerous times. So let's talk about it. In theory, grafting is quite simple. You just make a wound on an organism, and then you stick a tissue on the wound. Then you let it regenerate. However, grafting in most animals is difficult because there is a thing called anti-graft immune rejection response. That's why you need to be tested for blood or any other organ donation. If it's not compatible, it will be rejected by the body, and it can cause complications. Even so, there is one animal that is constantly being tested for grafting. That is axolotl. For most people, axolotl might be associated as pet. However, axolotl had been a laboratory animal since mid to late 19th century. That preceded the use of Drosophila as laboratory animal. While it was used for various research, the most important usage of axolotl as laboratory animal was for the study of Entwicklungsgeschichte, aka embryology. It was mostly done by German researchers. Starting from the 1900s, axolotls were especially known for their regeneration capability. They could regenerate without scarring, and they could even accept cells from other genotypes. And so, researchers started doing interesting stuff to them. That includes the parabiosis experiment that I showed in last week's video. To study its regenerative capability, and for the sake of transplantation research, allegedly at least, they started grafting. They mostly graft blastema cells from and to regenerating limbs. Oh, blastema cells are stem cells responsible for morphological regeneration, by the way. And yes, these graftings have a decent success rate. At some point, they even graft an entire head, which, at this point, is basically a parabiosis experiment, in my opinion. That also succeeds, by the way. Some of them, at least. The grafted head lives. Nowadays, grafting on axolotls are still being done for research in medical field. However, grafting in the current years are being done on embryos. With more sophisticated technology, they could do more specific research. The question of ethics on such research could be debatable. However, the case where axolotl grafting could mostly be viewed as unethical is the case where people are doing so to make it marketable. Let me introduce you to Firefly Axolotl. Firefly Axolotl was first created by Lloyd Strohl. He was doing a preliminary study into distribution and activation of melanocytes in axolotls, so he grafted axolotl embryos. The result is 6 Firefly axolotls. Because he basically doesn't need them anymore, he sold them. That was 8 years ago, maybe. And then, Firefly axolotl became a popular phenomenon for reptile enthusiasts. Even though axolotl is not a reptile, but it is what it is, I guess. The demand increases. People are asking for them, but Strohl did not make more. I mean, it was for the sake of research after all. And so, others started to imitate Strohl's work. However, most people are not capable of doing embryonic grafting. So what they are doing is the relatively easy way, skin grafting in adult axolotls. So when you see a grafted axolotl being sold as pet, that's what most likely happened to the axolotl. You decide for yourself whether that's ethical or not. There is another animal that we know exhibit tons of chimerism cases. That is, human. Probably because we diagnose a lot of humans though. Hence why we found many cases. In some cases, twins are fused together during embryonic stage, just like the case of half-sided Budgerigar. Pregnant women can sometimes receive cells from their baby and vice versa. That's called microchimerism. There was a case where a woman was banned from competing because she was diagnosed as intersex. Apparently, she doesn't have an extra chromosome, so not XXY, but she is a chimera. So she has some XX cells and some XY cells. Transplantation can also cause chimerism, especially bone marrow transplantation, even though it's technically compatible. The thing about chimerism in human is, most of the time, there is no morphological signs or symptoms. Some pigment-related condition can be caused by chimerism, but it's not always the cause, so it cannot be used for diagnosis. The only way to accurately diagnose whether someone is a chimera or not is by DNA analysis. So, who knows? Maybe even you are a chimera without ever knowing it. Another interesting concept is human-animal chimera. It's basically xenotransplantation between human and animal. 7 years ago, you might have heard of the human-pig chimera. That was the case where human stem cells were injected to pig embryos. In 2021, human stem cells were injected to monkey embryos. Both of these are allegedly done to study the early stage of embryo development and potentially for transplantation study. Is it the most effective and efficient way to do it though? That's debatable. In 2021, the concept of biocyborg was talked about and published. One of the enhancements mentioned in the article was artificial chimerism from various organisms. Yes, not just from animals, but somehow also plants, even bacteria and virus. That was an interesting idea, to say the least. You know, straight out of science fiction. It's an interesting read, so go check it out if you are interested. The article is open access, and you can just check my references in the description. Last thing before I wrap it up, I want to talk about Mosaicism, because you'll also hear that word if you are reading stuffs about chimerism. So, genetic mosaicism usually means a mutation that causes a cell to develop into different sets of cells with different genotypes. Emphasis on mutation. So, mosaicism can cause chimerism, because chimerism is simply the case where an individual has multiple sets of cells with different genotypes. Doesn't matter how. That should be quite clear. However, some people use the term mosaic and chimera interchangeably, and also mosaicism and chimerism, of course. I just want to clarify so you are not confused when you are looking into chimerism and mosaicism stuffs. And yeah, that was chimerism. Now you know that chimera exists naturally, and like I said, maybe you yourself are a chimera. Who knows what we'll learn in the future? Maybe there will be another unique case of chimerism, just like the case in Yellow Crazy Ant. But for now, let's just learn what is known. And that's all for now. Oh, by the way, if you haven't watched my last week's video on parabiosis, I suggest watching that, because parabiosis will cause chimerism. Anyway, enjoy your day.